Greetings, class. Today we're going to start chapter 5, which is titled The Working Cell. So this chapter carries on our pathway of going through the cell, what they look like, what they are composed of, the different organelles, and how they function together. But for this chapter, we're going to talk more about the plasma membranes, the types of proteins that are embedded within it, that help it with cell signaling, transport, stabilization. And then we'll also talk about how cells use membranes, their energy, and enzymes. So enzymes are a really big part of this chapter, and you may be familiar with them already, but if not, of course, we will be going over them. So let's look at our chapter 5 big ideas. We're going to talk about cell membrane structure and function. We're going to talk about energy and the different ways that our cells actually produce energy from the food that we eat. And then lastly, we'll talk about enzymes and how enzymes function and their basic definitions. So let's go ahead and head into membrane structure and function. So if you remember, membranes of the cell are composed of something called phospholipids. Looks kind of like a little soldier. So it has a phosphate head, and I'll put phosphate here. So a phosphate head, and then it has two little fatty acid tails, and I'll put Fa for fatty acid. Okay, so two little tails and a phosphate head. And this is important when we go through and try to describe the different characteristics of the plasma membrane. So the biologists use something called a fluid mosaic model to describe a membrane structure. So this membrane is composed of something called a bilipid layer. So we have two layers of phospholipids, so one little head facing towards the outside or extracellular environment, and then one little head facing toward the intracellular environment or inside the cell. So this is why it's bilayers, because remember, bi means two. So there's two layers of phospholipids. Okay, so the plasma membrane, this characteristic is something called selectively permeable or selective permeability, which means it only allows certain things into and also outside of the cell. And a really, really important part of this membrane structure are proteins. So let's go ahead and take a look at a membrane. Okay, so you see right here and right here, there's two separate cells. We have our bilipid or, bilayer of phospholipids right here. You see one layer pointing into the cell and then one layer pointing outside of the cell. Right here, these large purple structures, these are proteins. So they can either be peripheral proteins or more integral proteins. So these are integral because they are pointing to or attaching to or have contact with each side of the cell. So inside the cell and outside of the cell. We also have cholesterol, which is very important for stabilizing the cell membrane. Okay, so let's go through a few different things that you'll see, more of a close-up of the things you'll see embedded in this membrane. Okay, so for example, we have something called transport proteins. And if you take a close look at this picture right here, you'll see small substances going inside the cell. And then also small substances leaving the cell, and they are coming in and out through these different proteins. So one example is a channel protein, kind of just like a little hole in the membrane. Another one is an active transport protein. So anytime something is active, it has to have something called ATP, which is our body's main energy currency. All right, you also see some enzymes. Enzymes are proteins. So enzymes are always composed of a protein. So they are there to help carry out reactions to speed up the rate of reactions. Next, we have attachment proteins. So let's say, for example, we have microfilaments and other fibers here. So these actually attach to that extracellular matrix and the cytoskeleton to help support the cell membrane. Next up, we have receptor proteins. So they can either be signaling or binding to other solutes, or I can say to its specific things that they like. So for these types of proteins, they have something called specificity. So they bind to only certain molecules, and those certain molecules typically initiate some kind of change in the receptor, and then a signal gets sent down into the cell, and then something happens. Okay, so these signaling molecules, they're going to go ahead and bind here to these receptor proteins. The receptor proteins then relay that message by activating other molecules within the cell. We also have junction proteins. These are really here for attachments in between cells. And then lastly, we have glycoproteins. So kind of like what they sound, it is a protein with a branched sugar attached to it. So anytime you see the word glyco, or sometimes just GLY, this means sugar. Okay, so sugar attached to a protein. The major function of a glycoprotein is cell recognition. We all have very specific glycoproteins on every single one of our cells to make sure that our cells and our bodies are able to recognize our cells and not launch an immune response to attack them. Okay, so let's talk about some of the different ways that molecules are moved into and out of the cell. First off, we'll talk about passive transport. So with passive transport, it does not require energy. So no energy is required. The main way that these molecules move is something called a concentration gradient, where molecules are traveling from an area of higher concentration to an area of lower concentration. Okay, so for example here, for diffusion, we have a high concentration of dye on one side of this, we'll say this permeable membrane, and the other side has no dye. So what happens, these solid particles naturally diffuse across from this high concentration to the low concentration until it reaches something called equilibrium. So equilibrium, we can say there's typically the same amount of solid particles in each part of this solution. So equilibrium means there's no net movement of solid particles. And I'll put no net movement of solid particles from one side to the other because now everything has an equitable distribution of solids. Okay, so on the other hand, we have something called osmosis. So this can be a little confusing if you're just learning about the different types of passive transport. But osmosis is the movement of water. Remember, with diffusion, that was the movement of solid particles. But with osmosis, it is the movement of water across a selectively permeable membrane. Okay, so those are some of the key terms to look out for. Selectively permeable membrane and also the movement of water. Okay, so for example, we have, let's say, imagine two little test tubes. We have a lower concentration of solute right here on the left-hand side, and then a higher concentration of solute right here on the right-hand side. And in the middle, if you can see right here, there is a selectively permeable membrane, just meaning it only allows certain things to cross through. So after a while, the water is going to follow the higher concentration of solutes. So even though the water is leaving the side with the lower concentration, the amount of water molecules and solid particles are pretty much equitable. And now because the water has traveled over to the side of this membrane that has a higher concentration of solutes, now the concentration of water to solid particles is more equitable. Okay, so a kind of a key term to remember is that water loves to be around solid particles. It always follows solids. So a big example of this is something called salt. So just our normal table salt, something called sodium chloride. Water loves to follow salt. So maybe think if you eat a really salty meal, sometimes you retain fluids, sometimes people's hands get swollen or fingers, or sometimes even feet. It just depends. And that's just because the water is there, the body is retaining water in response to an increase in the amount of salt in the body. Okay, so now let's talk about tonicity. And this is the ability of a cell typically and its surrounding solution, and whether a cell will either gain water or lose water or just remain pretty much stable. Okay, so tonicity has to do with osmosis. So cells shrink in a hypertonic solution, something called crenation. Cells swell and then sometimes burst in a hypotonic solution, something called lysis. So lysis means to burst. And then for isotonic solutions, there's no net movement between the outside environment and the inside of the cell. So let's look at a picture, which might make it a little bit easier to look at and understand. So on the top here, we have an animal cell. This is a red blood cell. And on the bottom, we have a plant cell. And if you remember, there are a few key differences between animal and plant cells. One major one, if you can see right out here, is a cell wall. So animal cells do not have a cell wall for extra protection. So hypotonic solutions, water is going to flow inside the cell and then eventually burst. Okay, so why this is important, we'll say this solution out here has a lower amount of solid particles. I'm drawing solid particles. Let's say we have 4 out here. But let's say, 6, 7, 8. We have 8 particles on the inside. The water is going to flow from a lower concentration of solids to a higher concentration. And that is why all the water is flowing into the cell until it bursts. All right, nextly, we have isotonic solutions. So just no net movement. Everything is kind of normal and the same. For hypertonic solutions, meaning the outside environment has more solid particles than within the cell. We have a bunch out here, maybe 1 or 2 in here. That means the water is going to leave to follow those solid particles. And because the water is leaving to be around all the solids that it loves so much, the cell is going to shrink. Okay, and that's where you get this shrunken or crenated cell. It looks a little bit different in the plant cell, where here you don't get fully shrunken cells because the cell wall helps the cell from fully shrinking. Okay, it becomes something called plasmolyzed. All right, so next let's talk about those transport proteins. So these can actually function in something called facilitated diffusion. So hydrophobic substances can actually get through that cell membrane pretty easily because the inside, which is composed of those fatty acid tails on the phospholipids, those are hydrophobic. So anything that's hydrophobic or very small can easily slip through the plasma membrane. But on the other hand, anything that is polar or charged, okay, remember polar means having 2 separate charges, or just charge. These do not cross at all, or pretty much, not really easily or not at all. So instead, these things have to be helped across with the help of proteins. So this is called facilitated diffusion. This also does not require any energy, and it also relies on that concentration gradient. So transport proteins help specific substances to actually get across the cell membrane. The way that water gets through is something called an aquaporin, and this is a water channel. So remember, aqua means water, and think of a pore, kind of like a hole, right? So these are specific for water. And here's a picture. All right, so we talked about the different types of passive transport. So we have diffusion, we have osmosis, and then facilitated diffusion. Next, we'll talk about the kind of transport that does require energy. And anytime we're talking about energy expenditure or energy being used, we're referring to a molecule called ATP, which is also known as adenosine triphosphate. So in active transport, it does require at least 1 molecule of ATP to occur. Okay, so we do have a multi-step process. So for a transport protein, we have our solid particles, our solute is going to bind to the transport protein. ATP comes in and provides the energy needed for the protein to change shape. The protein is then returns to its normal shape and more solute can bind after it releases whatever this solute is into the extracellular environment. Okay, some more examples of active transport involve the movement of very large molecules either into the cell or out of the cell. If something is coming out of the cell, it's called exocytosis. If something is coming into the cell, it is something called endocytosis. So for both of these different processes, the material is transported and packaged in something called a vesicle. So kind of think of a little bubble that surrounds things, and then you have your solid particle right here in the middle, either a solid particle or it can be a liquid particle. So there are 3 different types of endocytosis. So the way things that are actually entering into the cell, we have phagocytosis, phage or phage, that means to eat. Okay, so this is the transport of solid particles inside the cell. We have pinocytosis, which is more of cell drinking, so engulfing of more liquid particles. And then lastly, we have receptor-mediated endocytosis. So this uses specific membrane receptors to bind to specific solutes. All right, so phagocytosis, they're the solid particles that we are trying to get inside this cell. It forms a little invagination that then pinches off and becomes a vesicle filled with this solid particle. For receptor-mediated endocytosis, these are our receptors, these little red Y-shaped things. They bind to very specific molecules. You see right here this purple triangle. They're binding to these receptors. A part of the membrane invaginates, and then it's going to pinch off and become something called a clathrin-coated vesicle that still has these specific receptors and the specific molecules now engulfed inside that vesicle. All right, so now let's talk about energy and the cell. So there are all different kinds of energy, and the cell is actually able to transform that energy and matter as they perform work. So kinetic energy, you might have heard of this before. It is the energy of motion. Potential energy is more of a stored energy. So think of maybe eating a piece of toast. What is stored in the toast? We have sugars, carbohydrates. So that energy is then stored in our bodies and then able to be converted into active or kinetic energy. Okay, so just a picture. So we take in fuel, glucose, which is the major molecule our body uses to make energy, and then also oxygen. This is what we breathe in. We convert that to something called ATP, remember our body's main energy currency. And then as waste products, we released carbon dioxide, which is CO2, and we also release water. Okay, so H2O. So let's talk about the basics of chemical reactions. They're either going to release or store energy. For exergonic reactions, these are going to release energy. Think of energy exiting an environment. Next, we have endergonic reactions. So think of energy actually entering into a reaction or into a space. So this is going to be one that has more types of potential energy, so more stored energy. Okay, so for example, we have our reactants. We go through a whole chemical reaction. Energy is released, and then you get products. Okay, so you have energy being released. For the next type of reaction, we have energy that's going into the cell. Okay, so we get our potential energy. All right, so we talked about ATP before, but this powers pretty much everything, all forms of work that our cell has to perform. So the way it does this is that we have this adenosine, and let me show you a picture. We have a molecule of adenosine, and then we have 1, 2, 3 phosphate molecules, right? So adenosine, tri. So tri means 3, and then phosphates, okay? What happens is we put a molecule of water in, something called a hydrolysis reaction, which we may have talked about before. So hydrolysis, it breaks apart a bond between the phosphates. So as this bond is broken between these phosphates, it releases energy. Okay, and that is how energy is released when these ATPs are actually broken up. Okay, so we have ATP, it's broken up, you get the energy for cellular work, and then it is reformed. So just a big circle. All right, last part of this lecture, let's talk about enzymes. So enzymes speed up a cell's chemical reactions by lowering activation energy. So enzymes are proteins, and they are something known as a biological catalyst, which means they decrease activation energy, they speed up chemical reactions, and they are not destroyed at the end of the reaction. So let's take a look. So we have a reactant, which is the first part of a chemical reaction, and it has to have some type of activation energy. The energy it needs, the cell needs to get started. It goes through the whole reaction, and then you get products. So reaction without an enzyme, it would take sometimes up to even years what enzymes can accomplish in just mere seconds. So with an enzyme, you see this little level right here, how much lower it was compared to this previous page, how the activation energy is so much higher. So enzymes will actually lower that activation energy and help to speed up our chemical reactions. Okay, so enzymes are something known as specific. They only bind to a specific substrate, and a substrate is whatever the enzyme likes to bind to. And this fits in something called an active site. So enzyme specificity is a term you might hear over and over again when we do refer to enzymes. So we're going to look at this next picture. So we'll say this is our enzyme right here, this purpley thing. And then it looks like we have an active site right here on the top of the enzyme. So this means the enzyme is available and is ready to do some work. The next slide shows that this substrate, something called sucrose. So sucrose is just regular white sugar you might get from the grocery store. It binds right here to the active site. So it's very specific. So this enzyme, which is sucrase, so typically, anytime you see a word ending in ASE, that means it is an enzyme. So sucrase binds to sucrose, which is our table sugar. All right, so it enters into our little active site. As the sucrose is here, this substrate is here, it is converted to products. So we have a chemical reaction occurring on the enzyme. All right, so after that chemical reaction occurs, the products are then released. So you have a glucose and a fructose. All right, so last subject about enzymes. So we have different things that can actually inhibit enzymes, and these help to regulate enzyme activity within a cell. So we have a few different things. We have competitive inhibitors, which means they compete with the substrate for the active site on an enzyme. We have non-competitive inhibitors, which actually changes or alters an enzyme's function by changing its shape. Lastly, we have something called feedback inhibition. And this happens, let's say in our previous example, that we had that sucrose, and we have the glucose and fructose after it's broken down. So this glucose will feed back to the starting enzyme and attach here so that no more sucrose can bind. And that just means we have enough glucose being produced in the body and we don't need any more. So that's an example of feedback inhibition. Your body's saying, hey, we have enough of this. We don't need to make any more for the moment. All right, so this is the end of Chapter 5. Don't forget to click below and make sure you take the Chapter 5 quiz.